In this video, we will discuss about FAK rule at different cellular local locations. So first, we will discuss it at the cell surface membrane, where the FAK gets activated through this extracellular matrix focal adhesions. So these focal adhesions interacts with the integrins that are present on the cell surface membrane. Then this will trigger intercellular signaling in a way that it will interact with taline, paxillins, and also this firm domain of this FAK kinase, and that will trigger these phosphorylation of the kinase, and it will promote the survival of the cells, proliferations, mechanical tensions, and also helps in the migrations. Whereas FAK in its own remains inactive unless it interacts with the integrins, which are triggered by focal adhesions through the integrins that are present on the cell surface membrane. And one more phenomena is observed, that endocytosis of these focal adhesions. In this case, cytoses are present. Cytoses can enter inside the cells, or these whole things can enter inside the cells in a way that now this in these endosomes, the FAK activations can cause suppression of the NUKIS. NUKIS means suppression of the program cell death through the activations of this FAK in the endosomes when there is whole focal adhesion kinases enters these endosomes. So in this case, this suppression of the NUKIS will occur. NUKIS means that program cell death. At the same time, it will promote, encourage independent growth, and this will also cause the autophosphorylations of this FAK, and this will help the cancer cells for the uncontrolled growth rate. And one more phenomena is observed in this of this FAK is about the somehow this FAK has been transported to the nucleus, and in this case it will helps in the tumor progression by degrading the p53. But how these FAKs are transported into the nucleus is not yet known. So these are three different locations FAK plays different roles. FAK on its own is inactive, but it gets active when the integrins interacts with the focal adhesions through the intercellular, through this cell surface integrins. And the activation occurs at these 397 phosphorylations at these 397 positions where Y amino acids are present. And this is the structure for, you can say that, three domains of inactive FAK, which gets activated when it interacts with the taline in the endosomes or intercellular parts, but when it interacts with the integrins on the cell surface, then it requires other components such as PI45, P2, taline paxillin, and RAB21. In together, this complex helps in this activations of FAK. So for this cancer stuff, this how this FAK transport into the nucleus needs to be identified so that we can target this tumor progressions. At the same time, since this RAB21 helps in the endosomes, we can target this RAB21 to prevent the endocytosis of these focal adhesions and thereby prevent the suppression of new cases and suppress this encourage independent growth. So these are two different kind of targeting approach can be used to diagnose or slow down this tumor progressions. So I hope you understood this FAK role at different cellular locations. That's all for this video. If you need to understand more about this topic, you can go through the reference. Thank you for your time.